okay, this one works. No, that's a very interesting question. So we're approaching that a number of different ways. It does appear that different participants, as you might imagine, have different amounts of antibodies. And we think that there are some people whom we would not have to augment with antibodies. But that's interesting, and that's part of the characterization that Harris and Brad are doing, because we're also interested to know whether the people with more of those antibodies have different reservoir sizes. And using the Cura assay, Brad has quantified now several hundred individuals for their reservoir size, and we have serum and PBMC stored on everyone, and so we can do that. The other thing is we're actually going back to HIVIG. You may remember HIVIG. This was sort of pooled immunoglobulin from HIV-infected donors that was tried some years ago, because it may be that that also could be an interesting alternative to antibodies. But we are negotiating with a number of people to provide clinical antibodies for a study that we are going to plan to do this year, as well as the CTL therapy study. So we will be doing a study where we hope to infuse a combination of antibodies, or one antibody plus Alt-803 in people this year in Washington.
So, Doug, I have a question about the CD4 CARs. Do they do anything to B cells or MHC class II expressing cells? Do they actually eliminate APCs?
We have not seen that, but we have not accessed a full range of tissues or been able to do that enough times to really say. And we are thinking potentially we'll have to use those CD4 CARs with other modalities as well.
No, but if you just, let's say, co-culture them with Ragi-cell or something, some other B cell transform B cells, do they kill them or not?
They do not.
Good afternoon. I'd like to ask Professor Stevenson, talking about macrophages as potential reservoirs of HIV, I'd like to know, do you think the macrophage have some kind of level of latency just like T cells, or they are reservoir through some other mechanism just like VCCs?
Yeah, that's an interesting question. We can, with macrophages in laboratory, we can reconstitute a latent infection. But that's using monocyte-derived macrophages that might not be true physiologic representations of, of macrophages in the tissues. We're now moving to these pluripotent stem cells, these iPSCs, to reconstitute microglia, because we think it's a more physiologic system to study latency. But from our in vitro studies, we can reconstitute latency. It's going to be hard to find out if it occurs in vivo, but people like Jake Estes are using RNA-DNA scope, which allows you to visualize cells, individual cells that are DNA positive and RNA positive. So one possibility is you could visualize infected cells that contain DNA but no RNA. It doesn't prove latency. Those genomes may be defective. But I think most of our information is going to be derived from in vitro studies. Now there's a model macrophage-only mouse, the mum mouse, where you only reconstitute macrophages. Those mice are highly infectable by HIV. All of them, the macrophages in the tissues get infected. And recent data suggests that the virus can be retained in those cells in the face of therapy. When you remove the therapy, the virus rebounds, suggesting there may be some latency going on. So so far it suggests that yes, there may be latency in those cells.
Thank you.
Hello, Dr. Stevenson, still on macrophage reservoirs. So if I understand right, one of the critics that people who don't believe in macrophages as reservoirs is that they don't live as long as memory CD4 T-cells. So I would like to know your opinion and whether you have any data showing that people who have been on heart for longer times might have less virus which recruit this from macrophages or something.
It's a very important question. You know, studies on microglia using whole body irradiation suggest that especially perivascular microglia have a lifespan of decades. So macrophages can live for a long time in the tissues, CNS for example. But what happens when they become infected is an open question. Now, a CD4 T-cell has a half-life of less than a day once it becomes infected because of cytopathicity. We don't see cytopathicity in macrophages, but there are a couple of studies suggesting that at least in the monkey model, that macrophages die quickly when they become infected. There are other studies with shivs in macaques suggesting that macrophages don't die when they become infected. So there's contradicting information out there on how long they survive once they become infected. Now, the problem with some of these studies is they're being done in models where CD4 cells have been depleted. And when that happens, the virus goes into almost all macrophages. And that's not really what happens in infected humans. And so the half-life that they're seeing when all the CD4 cells are depleted might reflect a rapid half-life in cells that don't normally become infected when there are CD4 T cells there. And that might be obfuscating, hiding what's happening in the long-lived macrophages such as microglia. So that's going to take a little bit of figuring out. But again, what we're doing to get at that issue is we have individuals who have undergone repeated treatment interruptions. So we're looking at the virus at those two interruptions to ask whether those macrophage tropic viruses come back, same viruses come back months later when the individual does a second treatment interruption. We don't have any data yet, but that's how we're going to get at that question.
Good afternoon. I have a question also for Professor Stevenson. I would like to know if you think there is some relationship about the CD4 density in different patients, in macrophages from different patients, and the differential selection of these macrophage tropic viruses in those patients.
So again, it's an interesting and insightful question because obviously in late stage AIDS, there is going to be a low number of CD4 T cells, which might create a very different environment for the virus. I mean, there appears to be more macrophage infection at later stages of AIDS, especially in the CNS. So the virus is forced to go into reservoirs because the target cell is rate limiting. We haven't done anything in that area, but it's quite likely that frequency of infection is dictated by the stage of disease and the number of CD4 T cells. But really, the important question right now is what's happening in individuals who are on therapy and whether these cells are really going to pose a problem to the sorts of elegant cure strategies that Doug just described. Because unfortunately, most of the focus has been on T cells, and for that reason, we really haven't come up with any ideas as to how we're going to deal with this macrophage reservoir if it does exist. Does that answer your question? No? Scopa. Yeah. It was something to do with CD4 density. I interpret that as CD4 cell density. Do you mean? Well, I mean, macrophages are highly heterogeneous. I mean, they're very different from tissue to tissue, and it's likely that the density of CD4 in the cells is also somewhat variable. But for the most part, CD4 is very limited on different macrophage populations, on Kupffer cells, on mucosal macrophages, on alveolar macrophages, on microglia. They all share the same characteristic in terms of CD4 density. So we haven't found any cell populations where there may be more CD4 that could perhaps allow T-cell tropic viruses to invade. We haven't encountered those conditions yet. Is that more in line with what you were asking? Okay, sorry.
So hello, my name is Vivian Avelino Silva. I'm from University of São Paulo, and I have a question for Doug. So Doug, we have some experience in lymphocyte infusion for bone marrow transplant recipients, for example, donor lymphocyte infusion, and that may be associated with quite a large, intense reaction, especially graft versus host disease. And similarly, in the situation of a lymphocyte infusion, such as the Believe Consortium is proposing, a very intense inflammatory reaction may also occur. And I think there's also the contrast of how convenient and easy and efficacious the current antiretroviral treatment is. So what do you guys predict that could happen in a potential clinical trial in the future, and also how to justify that in the face of the treatment we already have for HIV?
Thanks, Vivian. Important questions in relation to the safety profile. We are very cautious about this approach, but believe that if we collectively wish to find and eradicate HIV from an individual, it's going to require finding the cells that are infected. And our approach is autologous, so it's taking cells from someone and expanding them and putting them back. And that's very routine and very safe. So as I mentioned, Kath Bollard and many others around the United States does that on a weekly basis, minimal side effects. She has grown up CTL to CMV, Epstein-Barr virus, adenovirus, BKJC. She's also infused 3 patients with HIV-specific CTL with no deleterious effect. When we add other agents such as ALT-803, you're right, we might expect an inflammatory reaction, but that might be what we need. And we might need to have ALT-803 to upregulate CXCR5 to get them into the B-cell follicles where they're not getting to. So the concept is to try and push these cells into places where they might well cause an inflammatory reaction, obviously while maintaining antiretroviral therapy. So we're cautious, but we want to do something for people. And so we are going to do our best to go through the proper experimentations that also involve looking at non-human primates first to try and select what we're going to do. In relation to the question about why you would want to actually use this when we have such good antiretroviral drugs, I think it's amazing how antiretroviral drugs have, in combination, come to a stage where someone who can tolerate them can effectively do really well. But there still is a life expectancy gap. Even with the best antiretroviral drug regime, people will die before they should, and they will also potentially get non-age defining conditions, malignancies, heart disease, and then the stigma of becoming or being HIV positive. And when I talk to our community advisory board and our potential HIV participants, this is a main ongoing discussion. they do really tell me that they would like to participate in a cure study because the stigma being HIV infected is such that they would want to be at a stage where they would do. So those are the answers. The other thing is that it is a complicated system, but you know, the United States can be pretty expensive in medicine. But Kath Bollard has got this cost down to about $5,000 per participant. And so compared to treatment for hepatitis C virus, this is cheap.
Hi, I would like to ask a question to both doctors. I'm not a medical student, I'm just a system analyst. So it's very easy for me coming from the community to answer the question, what do we want as a community from the researchers? We want the cure. It's a very simple answer. But Dr. Nixon, you mentioned that the involvement of the community is very important to this project. And so I would like to ask you both, what would you say to this project in specific is what the community input is at most important to you? What do you expect from the community in terms of input, in terms of what we can contribute for you to achieve the goal?
I think I'm going to hand this over to Camilla.
Yeah. Eu acho que a participação da comunidade, desde o início da pesquisa, nessas primeiras fases, compreensão do que está sendo feito, a discussão, para se trazer justamente os questionamentos, quais são os riscos, quando, se existe possibilidade de cura ou não, tirar os estigmas que já se começou a questionar. Será que se tiver de fato a cura, as pessoas vão relaxar e não se importar mais com a transmissão, com a prevenção? Como vai ser todo o mecanismo de cura? E trazer também os anseios e receios que tem dentro das pessoas envolvidas e que são o alvo da pesquisa e do estudo, o objetivo final.
So, in addition, I think what we have decided to do in our collaboratory is to try and attempt something which is not often done, which is to have scientists like myself go to meetings where I have to explain in language which people who are not medical students or biologists can understand. But we are really learning a lot from the community in relation to the type of questions that Vivian was just asking me. Do people want to be enrolled in a cure study? If so, what are the ethical concerns that they have that we may not have thought about? So we have a very active community engagement group who meets every month, and they review our protocols. Each of our scientific programs has got a designated community member who we explain and talk and discuss what we're doing. We try and be completely transparent. We try and learn from what input we're getting, because that could well direct and dictate the way that we're going to do our study. So we find it very important. Here in Brazil, Esper and Camilla are developing a community advisory board. For people who are interested, they should be talking to them. And we are also working with our colleagues in Mexico and Canada. Our international community advisors are also really important, and each local site has different thoughts and different ideas. So we're excited about how we're working with the community.
Again, a very important topic. So in Miami, we have a very strong working relationship with several community organizations. One of the new programs that we're initiating is called Get to Zero, which is a bold plan to try and get the number of new infections down dramatically, because Miami is unfortunately number one in the United States. In Miami Beach, the infection rate is 157 times higher than the national average, which is remarkable. So we've been working with the community for two reasons. We need their advocacy. So you need to advocate for us, and we need community to keep it real for us, to tell us, you know, what is the reality and what we need to do. So for example, in the Get to Zero initiative, it's a collaboration between scientists, politicians, community, to come up with a functional strategy to try and get HIV infection down. It's not unique. There's a Get to Zero program in Baltimore, in Washington, D.C., New York, San Francisco, and in Hawaii. And all of those initiatives require strong relationships with community. So we need you for the advocacy and to keep it real for us.
Bom, gostaria de agradecer a participação, tanto do Dr. Mário Stevenson quanto do Dr. Douglas Nixon. Muito obrigado. E gostaria de...
Before we finish, I just would like to thank you very much for your excellent talks, and particularly Professor Stevenson in Portuguese. Never saw that before. Thank you very much.